All right. So, like I said, today we're not going to do that lecture on plant adaptations, even though that's what I had you kind of watch those videos in preparation for. We'll touch on that tomorrow. That being said, I believe we have groups doing the final project on passengers and stowaway in here. That lecture in particular contains some very good information that you might want to peruse, so I left it up in its entirety in case you want to take a look at that. But we're going to focus on statistical analyses this morning. And I'm actually going to end lecture at 12:30 today because I would like to attend this lecture, right? We have a former Gustie coming to campus, and you are all invited to attend as well. It's not required, but if you have interest in it. She will be speaking on plant physiology, specifically looking at hardiness, so winter hardiness and perennial ryegrass. So she does research at the University of Minnesota. So if you're interested, I encourage you to attend. It starts at 12:30. It's like opposite end of the building, apparently. So I'll likely end class around like 12:20-ish. All right. Another thing before we dive into stats here. I was reading through all of your responses to your lab post-lab document that you turned in yesterday. And a lot of you said, what would you do if this experiment was different? Right? You said you would plant like potato seeds or crops that could actually be eaten by astronauts, right? Awesome idea. I love it. If we could grow potatoes, I totally would. They just don't grow fast enough. But this interesting article came through my inbox today, actually. And it actually touched upon how these Chinese scientists have sent crop varieties into space, actually to the, I'm not going to pronounce it right, I know, but the Tiangong space station, so the Chinese space station. That was a space station that was only launched in 2021. It's smaller than the ISS, and operated entirely by China. But they launched potato seeds into space, all right, trying to see how they're going to fare with maybe mutations that might occur with the radiation hitting them. With the objective to, go in and breed plants that can be more tolerant to like salt and other saline alkaline soils that we might find, say, on the moon or on Mars. So, pretty interesting experiment already happening. Thought I would share that with class today. All right, but like I said, we're going to do a crash course in stats, specifically looking at ANOVA. If you've had stats before, right, if you are a statistics major, this is going to not go into too much depth. It's very much surface level stats. It's probably going to be missing a lot of what you might want in a stats crash course. But for those of you who have never had stats, right, this is going to give you the basics and the introduction to know what we're talking about when we are talking about running statistical analyses. All right, so first of all, why do we need stats, right? Well, stats is a great way, statistical analyses are a great way to state how unlikely our results are to reflect some chance process, right? So we run a bunch of different replicates, right? We analyze them, and we say, yes, it's different, and it's significantly different. It's not different just due to random chance, right? Maybe the 5 plants we looked at, right, all grew the same way, and that was by chance, right? And so statistics is going to tell us how unlikely our results are to reflect this chance process, right? And remember that science can never prove, right? We can only provide support to our hypotheses, right? So statistical analyses really gives us the confidence in saying the likelihood of something, right? Saying that this is different, right? Or this is likely not different between our treatments. All right, so if we look at this graph here, right, we are graphing over 4 different months, right, and the days of snow, right, so days that actually snowed. And if we looked at this data, right, would you say the months are different from one another? Just by looking at it? Yeah, I would say so. How confident are you on saying that they're different? Yeah? No? Maybe? Somewhat? Yeah, I mean, it looks pretty much like, yeah, February has a lot of snow, and that's very different, right? Well, stats are going to give us some data and the confidence to say that, yes, it looks like February has a lot of snow, right? And my statistical analysis has also backed up that by saying that, yes, there's a significant difference between my months, right? So that's why we run stats, to give us the confidence in saying, yes, something is different. This is not just a chance event. But if we think about what data we're inputting, right, we really have to pay attention to sample size here. And that's going to come into play with our data as well. So if we look at December, January, February, March, right, if I looked at 2 years worth of data, right, is that going to be more or less informative than, say, 10 years worth of data? It's likely going to be less informative, right? Because I only have 2 years of data going into this analysis. Versus if I have 10, that's going to say that those 2 years were likely not by chance, right? I have more data to support it, right? Think about if we entered in our data for this year, days of snow, all right? It's going to look very different from other years, all right? So data is variable. That's why we want to collect a lot of different replicates, which is why I had us all grow basically our own experiment for the growth chambers, right? So we had 7 groups. Theoretically, we should have 7 replicates, right, for each treatment. But we know that's not the case, right? That Martian soil was harsh on our plants. And in one, I think it's just the Martian without compost, we only had 1 plant survive, right? So even though we could have had 7 replicates, for that treatment, we only have 1 replicate, right? So replicates is something to pay attention to, especially when you're writing your report today. So if I had 10 years worth of data versus 1 or 2 years worth of data, right, it could tell me something very different and give me more or less confidence in my results. All right. When we analyze our data, we often choose this threshold, right? An acceptance or rejection probability. We can call this the alpha value, right? Make sure we don't mistake variation from significant biological variation. So there's variation in nature and everything that we study. Nothing's going to be perfect, right? And so we're going to set a threshold to say if it falls above or below this threshold, we can either say that there is a difference or there isn't a difference, right? Or how unlikely does it have to be that a difference would occur by chance before we are convinced that it is real, right? Now, in most sciences, the threshold is going to be set at 0.05, saying there's a 5% chance that this is, or 5% chance that this is due to random chance. 95% of the time, though, it's not due to random chance, right? So we would expect to see a difference by chance only 5% of the time. Now, if you look at maybe, say, like the medical field or maybe even the physics field, right, where we have to get even more precise measurements and like medical devices need to be very, very accurate, right? That threshold might change, right? And in medical devices in particular, I think they often set the threshold to 0.001%, right? So they want to see a difference by chance, like less than 1% of the time, right? Because medical devices, think about like a pacemaker, you don't want that to fail on you. All right, we are specifically going to do an ANOVA today, right? And because I am not the best person to talk about stats, right? I am not a stats major. I know very basics of stats, right? I know a lot more ecological statistics than flat out mathematical statistics. This video does a really nice job of kind of explaining what an ANOVA is, right, and what we can determine based off of an ANOVA analysis. An ANOVA is exactly the type of analysis that I have done to our data, and I will share those results with you shortly.
What is an ANOVA? An ANOVA is an analysis of variance. It is an extension of a t-test. While a t-test determines if 2 group means are significantly different, an ANOVA allows you to determine if significant differences exist between 2 or more group means. For example, you can compare mean test scores of students in private and public schools, as well as students that are homeschooled. An ANOVA assumes your data follows a normal distribution and that the variation within the groups of interest are equal. For an ANOVA, there is a null and alternative hypotheses. The null hypothesis states there is no difference between the group means. For example, no difference exists in the mean test scores between children studying at home or at a private or public school. The alternative hypothesis states there is a difference in the mean test scores between 3 school systems. The value resulting from an ANOVA is a p-value, and it tells us how likely the difference between the groups could have happened by accident. If the p-value is less than 0.05, then we know a statistically significant difference exists between groups, but we do not know which groups are statistically different from each other. For example, when comparing the mean test scores between the 3 school systems, a significant p-value will indicate the mean test scores between the school systems are statistically different, but not whether groups are statistically different from each other. In this case, a post hoc test is used. Post hoc is Latin for after the event. It is a test you would run after you've completed an ANOVA with a significant p-value. Post hoc tests are used to determine where the differences occurred between groups. For example, the post hoc will test if a significant difference exists between private and public school students, public and homeschooled, and private and homeschooled. Remember, performing an ANOVA is best practice when you have 2 or more groups of data, rather than running individual t-tests for each group comparison right away.
All right, so an ANOVA is what we will be looking at today, and like the video states, right, it compares the means of 3 or more groups, I guess 2 or more groups, so if you have 2, I don't know why you wouldn't run a t-test, besides the point here. Comparing the means between 3 or more groups. Now we have means, right? We have the average growth rate for each of our treatments, right? Martian, and compost, earthen, and compost, right? So we actually have 4 treatments. And so we are going to run an ANOVA to see, is there a difference between my treatments, right? Now I'm not going to know which treatment is different from which, right? I would have to run a post hoc test like they explained in the video. I have not done that simply because I don't have the statistical software on my computer, right? But we will take a look at the data to see maybe what values are driving the results. The ANOVA is going to give us a p-value. All right, that p-value is very informative in saying whether or not it's a chance process, or whether or not we can reject or fail to reject the null hypothesis, right? Again, notice that terminology there, right? We're either rejecting the null hypothesis or we're failing to reject the null hypothesis, right? If we're failing to reject that null hypothesis, it's like saying we're accepting the null hypothesis, right? But again, in science, we don't want to just flat out accept anything, right? So instead, we're going to say fail to reject, right? Kind of a strange wording in a sense here. Don't let that confuse you. But like I said, it all comes down to this p-value, right? So the p-value, if it's greater than our threshold of 0.05, we can fail to reject the null, right? Which means that we will accept the null in saying that there is no difference between our samples, right? Or if it's less than 0.05, we can say we will reject the null, meaning there is a difference, right? Obviously, most of the time, we hope to see that the p-value is less than 0.05 because that means we have significantly different different responses from our different treatments, right? And so I have run an ANOVA. This is not our actual data here. But this is just showing you how to read the results of the ANOVA test, right? I ran an ANOVA test on Excel, right? Which is why I did it myself, because I know not all of you have Excel. You have to download specific tools for Excel to be run. And honestly, Excel is not the best statistical tool out there, right? But it's one that's the most easily accessible by everybody, right? So I ran an ANOVA test. Yes? Yeah, they do. They do. I just don't have it on my computer. I'd have to use a college computer or virtual lab. And virtual lab is not great. Yeah. So I did it through Excel. You can do like a t-test, very basic statistical analyses through Excel. You can't do a post hoc test as far as I'm aware of, which is why I did not run one. right? Well, the results would look something similar to this. So this is last year's results, actually, when they looked at Martian, lunar, and earthen soils, right? It gives us a lot of information, but what you really want to focus on is this p-value here, right? So this p-value was 1.45134, all right, to the negative 5, all right? So it's actually like, with 5 zeros in front of that 1, all right? So it's 0.0000. It's very much below 0.05, right? It's a very negative value, which means there was a difference between the growth rate of those 3 treatments that they ran last year, all right? All right, I will show you the results of our ANOVA, and I'm actually going to have you interpret the p-value yourself, so that's why I'm holding off on telling you the results, all right? Today in class, the next hour-ish here, I want to leave a little bit of time for the discussion at the end of class here. We're going to work on graphing your data, right, interpreting the data, and getting all the figures ready for your report, all right? So when we graph our data, right, we often want to look at the type of data that we're graphing, right? We want to see if our variables, remember we have dependent and independent variables, right? They can either be discrete or continuous, right? Discrete means they fall into a category, like blue, green, orange, right? Think about our variables, our independent variables, our soil types. Those would be a discrete variable, right? They fall into Martian soil, earthen soil, right? Or one of those with compost. Whether a continuous variable is going to have a spectrum of numbers, essentially. So like 1.1, 1.2, 1.3, 1.4 can have a whole range of values, right? Our growth rate data, right, is an example of a continuous variable, right? We have variable growth rates ranging from less than a centimeter to a day to, I think, did we get over 2 centimeters a day? I'd have to double check that, right? But it's a range of values, right? And when we're thinking about graphing our data, right, this is going to tell us what type of graph to use, as I'll say on the next slide here. But the graph should really be standalone, right? If I'm looking at a graph, right, I am not going to read the whole paper. I'm going to look at my graph. I'm going to look at my figure caption. I'm going to know everything that I need to know, right? Now, if I want to know more than that, I can go and read the rest of the paper, right? So in our graph here, I skipped a slide, right? I'm doing a bar graph, right? Because our x variable, or the variable along the bottom, is categorical, so our soil types, right? And our y variable, right, or the variable along the vertical axis here, is continuous, which would be our growth rate, right? And then below our figure caption, or below our figure, we're always going to have that figure caption, right? So that is why we have done a bar graph for our data. And again, I made this bar graph for you simply because I know not everybody has Excel. Yes, I know Google Sheets can make a bar graph, but they cannot customize the error bars, right? Which is why I had to make it in Excel. If you want to make your own, by all means, the instructions are in your lab manual. You can make your own. I tried to simplify things for you, right? So error bars, like I said, Google Sheets cannot customize our error bars, and we want to show the error bars because it measures how variable our measurements have been, right? So our measurements are very variable. Some of your Martian plants grew almost as tall as, or taller, than your plants that grew in the earthen soil, right? Others of you saw opposite results, right? There's variance there. And those error bars are really graphing and showing a graphical representation of the variance that we might see, right? Now we're going to graph the standard error, right? And it's often necessary in your figure caption to say that the error bars represent standard error, because sometimes the error bars can represent standard deviation, right? But we're graphing standard error for a variety of reasons, right? So basically, it can kind of give us a sense of how accurate the mean of any sample is. So if we look at this figure here that I have It's measuring two different techniques on measuring the mean oxygen saturation of blood, right? So do two different techniques, right? They gathered some data, they put it into a graph here, and we can look at the results. So technique 1 here, right? And technique 2, their means are very close to one another, right? But in technique 1 here, that error bar is very short, meaning the variance surrounding how we got to that mean, right? All those numbers used to calculate our mean, right, are more concentrated around that mean number, rather than technique number 2 has this very large error bar, meaning that the data used to calculate that mean were much more scattered around that mean, right? So it can tell us something about our data. All right, I'm also going to have you put in a table. All right, now you can choose what you would like to put in your table. You can focus on germination data. You can focus on flowering data. If you find a nice, concise way to put them both into a table, great. I'm going to let you choose which data you think is interesting and most important to represent in a table, right? And I'm going to have you make a table to put in your lab report, right? Now, I like this slide because I kind of put in some things that I would be looking for a nice scientific table, right? First of all, I do have a figure caption, I guess table caption here. It goes above the table, right? And it simply states what I'm looking at, right? The table itself, right? I like the independent variable in the far left column, right? So the first column that I'm looking at. In this case, we're looking at the age of different lizards, right? And I don't like having horizontal lines, right? I think it just makes it a little bit more distracting. I like very clean looking table. That's very easy to read, right? So when you're making your table, I would say, look back on this slide, rely on this slide. All right, like I said, crash course in stats, very much a crash course. For your paper, right, I want you to write a figure caption for the average growth rate figure, right? I want you to make a table of either flowering data or germination data. And I want you to be very mindful of sample sizes, like we talked about before. That Martian soil only had 1 for a lot of the samples, right? Versus our earthen soils all had 7 as a sample size. All right. I've given you kind of guidelines for how to formulate these figure captions and table captions. But like I said, I'm going to have you all work on it in class here so that you can directly ask me questions and not have to wonder, should I include that? Is this better than this wording? So on and so forth, right? Figure captions, they're going to go below a figure. You haven't written one before, but you've filled in the blanks for one before. You can use that as a template again for this experiment, but tailor it to this experiment, right? The first sentence is going to be an incomplete sentence, right? And it's going to tell the reader what your variables are, right? What's your independent variable? What's your dependent variable? What organism were you studying, right? And what were the sample sizes? And the remaining sentences, they will be complete sentences, and they're going to tell the reader a broad overview of the experiment. So how did you obtain your data, right? Now, I'm not looking for very detailed numbers like I measured the soil or I planted the seed a centimeter under the soil, right? That's not necessary here, right? That would be for the reader to look into your method section. Instead, I'm looking for something along the lines of, we did a growth chamber experiment for X amount of days and measured this daily, right? You also want to include the trends seen, right? So a couple of sentences saying, well, it looks like our Martian soil plant, right, did not do so well, right? Or maybe the other one did a lot better or twice as well as the other, right? Some sentence telling me that overall trend. And I want you to include the statistical test performed, right? So I want you to say that ANOVA was run, this was the threshold value, this was the p-value that we got, all right? Again, I'm giving you a lot of information, which is why I'm going to have you work on it in class rather than out of class, so I'm here to help you along the way. right? Table caption, we kind of already went over that. I will let you read that on your own here, right? But this is our data. This is the results of our ANOVA analysis here, right? So our p-value is down here. I want you to interpret what our p-value means, right? I'm going to have you do that as a team, right? Next slide here has our graph, which you can copy and paste into your paper here, right? And you will need to include the figure caption below the graph. All right. I'm going to change the homework slide, right? This is the homework for tomorrow, but for the next little less than an hour here, I want us to work on making that table and making that figure caption, right? Asking me questions. If you get done early, you think you have it, you want to step out and go get some food, by all means. I'll say we'll meet back here at, let's meet back at 11:50, all right? But the rest of the time is work time, time for you to get some stuff done for your report with me in the room.